Let's get started. I hope you had a nice break. I'm a good lecturer. I'm ill during the break. Anyway, we are back, and isn't it a beautiful day to be back here in the L Theater? We will talk more about experiments today, so we will follow up on where we ended before the break. We'll get a little bit more into experiments before then. Next week we get into sampling a little more, selection bias, and then we actually start with analysis of data. Before then, afterwards, we come back to some more qualitative ways of doing research. Anyway, as a little side note, next week, not next week, on the 18th of April, we're going to have folks from RTE coming here. Right? Well, I give you another warning. Actually, we are involved in a study right now, the School of Sociology. I'm sort of partly involved in this. And we will show you some research that is happening there, which is actually about you guys, which is about the young generation. Anyway, but coming back to experiments. So today, before we start, it's the time again. The next assignment is actually online. I put it up on Blackboard yesterday. You find it as usual, and next week you will have your tutorial groups again. Another thing that a bunch of people asked me is about the exam. The time is coming. Still a few weeks, but we're getting there. Eventually, there will be an exam. There will be an exam about this. There will be questions. There will be difficult questions. There will be easy questions. I don't know. Depends on how much you prepare for this. And what I was asked and what I actually put up now on Blackboard, you will find a test exam on Blackboard. So when you go on assignments, you will find a test exam on Blackboard. So that exam will help you. So I highly recommend to have a look at this to get an idea about the type of exam, how this will look like, right? So the actual exam, the format of the actual exam will be very similar. Actually, instead of 7 questions, there will be 6 questions where you have to choose 4. But the other things are exactly the same, more or less. The questions, I don't know. It could be that some of the questions from the test exam are in the final exam. It could be that none of the questions from the test exam are in the final exam. I don't know. Don't ask me. Okay, so that's a test exam if you have for those of you who had some questions about how the exam will look like. Because I'm aware this is the first time that this course is actually being taught, so you won't have access to previous exams. So this is one sort of previous exam. Okay, let's get started with experiments. So today I want to talk about we already had experiments. It came up during our research design session, and then last time it came up and I showed you some examples of experiments, but now we'll have a more thorough look at that. And then we will look at different experimental designs and what we mean by that. Then I will talk a little bit about what we call Hawthorne effects. These are sort of effects where the experimenter, just by doing the experiment, already biases the result. So once people kind of discovered that, I think, hang on a moment, experiments are really great, and I'm a big fan of experiments. They are really great, especially when it comes to things like causality, but you still need to be very careful, especially thinking about what is your impact as a researcher on your subjects, on the people that you're studying. Right? So Hawthorne effects, we'll come back to that, was actually that people noticed that by doing a study, by being part of a study, people behave differently. So it was nothing about the treatment that people thought that they gave to their subject, but it was about being part of a study to begin with, which sort of seemed to drive the results. And then at the end, I'm going to talk about 3 different types of experiments. So we'll talk about laboratory experiments, which is probably what you associate with experiments, but then hopefully, especially based on the last lecture, you see there are other types of experiments that we can do as well. So there are field experiments. Remember the broken window stuff? That was pretty much field experiments. And then the last one is a fascinating thing as well, that actually I'm conducting some work on right now. It's what we call quasi-experiments or natural experiments. It's not a real experiment, but it looks almost like an experiment, and we exploit some random shock or something outside that kind of gives us the conditions like in a normal experiment. Anyway, so let me get started with experiments. So experiments, there are different kinds of research that we can do. Remember in one of the very first lectures, I talked to you about, some people, they say all we can do is interpret things. I don't believe that. I'm very much a big fan of really trying to understand why does the world work like it works. That's kind of really what I think what we should do. Some other people think we need to describe stuff, and that's all we do as sociologists. Well, I think description is extremely important, especially when you want to get towards understanding the world at the end of the day. You need to describe it properly first, but at the end, we still have this thing, what we call explanatory research. We want to explain stuff. Or at least that's what I want to do. I want to explain stuff. I want to understand why things are the way they are. And we want to get to this point of that and understanding the mechanisms, how the different elements sort of are all integrated with each other and how they eventually produce the outcome that we observe. Okay. Now remember, also in one of the first sessions, we talked about correlation and causation. Coming back to these kind of things. And there we observed that sometimes things seem to look as if they are related with each other, but when we look closer, they're actually not. Right? Could be a spurious relationship, could be something else that is driving the results. Right? It could be coincidence, it just happens by chance. Sometimes the stars align, and this is what we observe. When we want to explain stuff, we want to get to causality. That's sort of the thing. What is sort of causing the phenomena that we observe. So this is just back the slide again of what we have causation, that there it's clear that something implies something else. Right? Something causes something else. And that's sort of the holy grail in social science research. We want to understand why the thing happen as they happen. Okay, now experiments come into play, because experiments are really good for that. Experiments are really good for getting towards causality. Actually, they are excellent when you have a theory with a hypothesis and you want to test a hypothesis. They're excellent for explanatory research, and they're also good for small group interactions. However, remember last time actually I showed you that you now can use experiments on a much, much larger scale as well. Right? Nowadays people do online experiments with thousands of people. Actually, there was one paper where they did a Facebook experiment where they had 65 million Facebook users. They exposed them to a treatment. Facebook was on board with that. That's kind of pretty cool. And then, it was not kind of a small group of people or a small group of students or a group like you, which is actually already very large for an experiment. But, we can go all the way up. So there are all new possibilities. Traditionally, historically, laboratory experiments were much more focused on small groups. But nowadays, that does not necessarily hold anymore. So what's the idea behind an experiment? Well, the idea is, in contrast to simply observing something, right, or looking at variation in the population, let's say, I don't know, what is the performance of students who attend the lecture and students who don't attend the lecture at the end of the day, right? I could collect that data. But there I'm not taking some action. In an experiment, I'm taking some action. Let's say I force some students to attend the lecture and I force others not to attend the lecture. That would be sort of the action that I take. Experiments, we take action and then we observe the consequences of that action. And actually we call that action treatment. So at the end, it's as simple as that. You give some people a blue pill, some people a red pill. Right? And this idea, that might be obvious to you when it comes to, medical research, for example. That's the idea we can use in the social sciences. We expose some of our individuals to a treatment, and we have the control group, and we have the out-of-control group. Yeah? And then we're comparing them with each other. Remember the little survey experiment that I did with you guys? I separated the lecture theater in two halves. I gave one group the questions slightly differently worded than in the other one. Then I observed the differences. And that's the basic idea in an experiment, that we kind of split up our population in a treatment and in a control group. We administer the treatment, and then we observe the differences between the two groups. Well, of course, there's some more to it. So, for example, we can often what we do, we pre-test both groups, the treatment and the control group. Let's say I look at coming back to this example of whether attendance actually increases your grade at the end of the course. I could go and measure your exam results at the end of the day, but I could also test you before, right? I could have, before the whole thing starts, I have an IQ test with everybody of you, right? And then I kind of look at the changes. Then I look at how the changes in the control group from one time to the other differ from the changes from the treatment group from one time to the other. Yeah? That would be what we call a pre-test, post-test design. But we come back to that later. Okay, essentially the key is really the computation of the differences of our treatment and our control group. That's all there is, right? It's not overly fancy, but yet extremely, extremely powerful. Because the idea is here then that all those other things that matter in the world, all those other things about you that I don't know and that I probably shouldn't know, all those things, here the idea is, actually that's unobserved heterogeneity. There's also stuff where you are heterogeneous in a certain way and it's unobserved. I don't know it. I don't have the data. I don't have the information. Maybe you don't have it. I don't know. But the idea is that this unobserved heterogeneity, when I split you guys in two random groups, completely by chance, that all the things that I don't observe, they should matter equally for the random group, for the control group, and for the treatment group. That's the idea. There's a whole bunch of stuff that I don't observe, but the idea is that these things that I don't observe, they matter equally for the treatment and for the control group. That's the essence of an experiment. And you see how powerful that can be. Because that means I do not necessarily have to observe everything about you. Actually, I'm only interested in the treatment. Right? And that kind of gives you a much stronger leverage than when I do an analysis where I depend on variation within the population. Because then I don't know. Maybe certain attributes that you have, they correlate. Maybe, I don't know, the guys that come to the lectures here, they're just more interested in the whole thing to begin with, right? And that's sort of why they are, at the end, study harder, and this is sort of why at the end they also do better in the exam. Right? So these are the things, that's often the problem with studies where we don't have an experimental design, but where we rely on variance within our population. So in experiments, the key is that all the things that we don't observe, that's what we call unobserved heterogeneity, matters equally for the treatment and for the control group. So that's sort of why we also call this, this is sort of the gold standard. Gold standard. This sort of gives us really strong evidence for the things that actually happen. Actually, we call it also internal validity, high on internal validity. I'll come back to that later. Basically, it means that what we can be very confident that what we actually show really is happening. That it's not a fluke, because we changed something, we manipulated the treatment, we administered the treatment. So the differences that we do observe, if there are any differences at the end of the day, remember, I looked at all the differences in how much you agree with the water charges, things like that. Yeah. If there are any differences at the end of this day, it must be due to the treatment. Because everything else matters equally for both of the groups. Okay, so there we are already with random assignment. Now, random assignment is an important aspect or an element of experiments, and we'll come back to that next week when we talk about selection bias and randomness in general. Yeah, like what is randomness? Yeah, it's actually not that easy. It's actually not that trivial to define what randomness is. But with a random assignment, we basically mean a method of assigning cases. This could be individuals, could be organizations, could be groups, for the purpose of making comparisons. Right? We distinguish different groups, and we generate two groups that do not differ in a systematic way. That's the idea. Yeah, I randomly assign you to the treatment, to the control group. To be absolutely clear, actually, when I did this experiment with some of you guys sitting on this side, sitting on that side, assigning you to the different groups, I don't know, maybe there's something about people preferring to sit on the right side of the lecture theater. Maybe they think more with their right side of the brain, and maybe this is sort of more analytical or whatnot, right? So these things actually could be the case. It would be even stronger if, as you walk in randomly, I randomly assign you a number and I randomly assign you into one of the groups, right? But there you already see how the practical things kind of matter. How would I do that? It would be utter confusion. I don't know, even having that and kind of splitting up the lecture theater. 10% of you guys didn't assign yourself to the wrong group. I asked this other question before, whether you are in the black or in the white group. So these things are actually not that trivial, how we actually randomly assign individuals. The idea is, as I already mentioned before, through this random assignment, we have this leverage. We can say that we have an equal likelihood for being in the treatment and the control group. All these other things that we don't observe also have an equal likelihood for showing up in the control and in the treatment group. And then the individual differences, they cancel out. So here's an example of a random assignment. Imagine a whole bunch of individuals. I just displayed them here as circles. And they have different colors. The colors now stand for different attributes that they might have. It could be age, it could be sex, whatever. I don't care. It's just different attributes that individuals here have. And now a random assignment would be, I split it up into two groups, and then these colors have an equal chance to show up in both of these groups. Right? It's just really randomly putting things to the side. Actually, it could still happen that, I don't know, all the yellow guys end up in group 1 and all the blue guys end up in group 2. Yeah? Actually, there are only two group 2s. Anyway, you get the point, right? They're kind of two groups. It could still happen. It's just very unlikely, right? But that's kind of stuff we will talk about next week when we talk about randomness, because sometimes a lot of things cluster together, happen. You think, this can't be randomness, this can't be chance, but actually it is. Yeah? A lot of things just happen by chance, right? And that's sort of something that our brain is not really good in comprehending. We always tend to see meaning behind things. Yeah? We're sort of evolutionarily trained. We are meaning-making machines. That's sort of what we are. We're trying to find some heuristics. What are the guiding principles that produce the things, right? I don't know. Imagine, a few hundred years ago, people couldn't understand why is there suddenly an eclipse? Why is the sun getting dark? They thought they must have done something wrong. They must have annoyed their gods, right? People looking for explanations for things. But sometimes it just will happen. It just will happen by chance. But we will talk more about that next week. It's a fascinating topic when we talk about chance and what randomness actually is. Anyway, so this is sort of a random assignment, right? If you would do this often times enough, if you do it many times, then you would see that sort of these colors seem to be equally distributed in the different groups. While if you have individuals choosing their groups, it could end up that individuals with certain attributes end up in the one group, while individuals with other attributes end up in the other group, right? And that's what we would call a systematic error. So that's sort of why, in these experiments, we don't let individuals choose their own groups. We tell them, we assign individuals to groups. And to a certain degree, that's what I try to do, right? I said, you guys over here into this one group, and you guys over there in the other group. At the end, I still had twice as many people in the white group than in the black group, although they were equally distributed over here. So that means some of you must have lied. So that's sort of the thing that I kind of can see here. So that would be a systematic error. when individuals, or it can lead to systematic error, when individuals assign themselves into groups. So that's sort of why we try to do that as an experimenter, as a researcher. However, there's another problem, and we'll come back to that in a second when we talk about Hawthorne effects. By assigning you guys into a group, to the random or the treatment group, by knowing to which group that you guys belong, I might behave differently to the people who are in the treatment group. And actually, that's what we observed in many studies, right? Some people were given the placebo pill, and other people were given the real pill, and then doctors treated them differently afterwards. Knowing that they are in the good group, they should, they should notice the benefits of the drug because they got the real drug, while in contrast, they behave differently to people who got the placebo. And then at the end, what was the result? That people who got the real treatment, they sort of improved their health, for example, over the individuals who were in the placebo group, but it had nothing to do with getting the treatment, with getting the magic pill compared to the placebo pill. It had all to do with the researcher or with the doctors who helped them through this whole study, treating them differently, because they knew they are in the treatment group. Right? So you see how actually these problems kind of can factor in here by participants, by people knowing to which group they belong, or by the researcher knowing to which groups that participants belong. The solution to that, and that's something we come back to, that is the double-blind experiment, where neither you know nor I know who is in the random or who's in the treatment and who's in the control group. But before we get to that, let's talk about experimental designs. So there are a whole bunch of different experimental designs. There were several in the readings that you guys had to do. In the Bhattacharjee text, there were sort of very detailed designs, and then you kind of see how things can differ. And when you do experimental studies, you need to dig deeper into that. You really need to understand what are sort of the designs that matter, and what are designs, what they can do and what they cannot do. For now, what I'm going to do, I'm going to show you different kinds of designs, and I'm going to talk a little bit about their strengths and their weaknesses. So let's start with the first one. This is probably the one that you might have associated with the experiment to begin with. Yeah? There's sort of a group of people, I do something with them, and then I observe them. Yeah? Imagine the example, a man who exercises is observed to be in good shape. So that's what I'm observing. What do I know? Remember from the lecture that we had about causation, coincidence, and so on, actually when we observe that this guy is in good shape, we cannot really say that exercise helps, that exercise makes them healthy. Why? Because we have nothing to compare it with. Right? Maybe everybody is just in good shape. Right? So we need to have some comparison. And that's sort of what these experimental designs are all about. It's all about what are the comparisons that we use to draw conclusions about what we find. So when we just observe that, somebody who exercises a lot is in good shape, we don't know anything. Unless we compare it, and that's sort of what the one-shot case study does, unless we compare it with some intuitive standard of what constitutes a trim or good shape. Right? So we have an idea in the background a little bit about, what is healthy, what is sort of a good shape, and that's what we would have to compare it with. But strictly speaking, we don't have that information. We don't have that information. That's sort of where other designs come into play. So that's actually why this design is actually a pretty bad one. That's sort of a pretty bad one because we don't compare our what we observe at the end of the day, what we call our dependent variable. That's how healthy or how fit is somebody. We don't really compare that to a solid measure. Well, a better way would be what we call the one-group pre-test, post-test design. Okay, let's walk through this slowly. Why is it one group? We're just looking at one group. Now, I don't have a control or a treatment group here. I just have basically the same individuals at different points in time. Right? I have sort of individuals. I observe them at time 1. They sit on the sofa, they sit on the couch. Then I know that at time 2, they do exercise. and then I observe them at time 3. Right? So now I can actually compare the shape of an individual at time 3 compared with the shape of this exact same individual at time 1 before the guy did some exercises. Right? So now I have sort of created my two groups artificially here. Right? Like the one is before the treatment was sort of administered like the exercise, and one is afterwards. So artificially I can separate them into two groups, I can compare the different groups, and so on and so forth. The big advantage here is, think about some individuals just are in better shape than others. Right? By comparing the same individual with himself or with herself, I can rule these things out. Right? Unless now there's some other factors coming into play, maybe time factors in, maybe just people degrade after time. But let's say I do this in a relatively short period of time, then I know, it's the same individual that I have at time 1 and at time 3. The only thing that is different is that in between, at time 2, there was the exercise that happened. So that's sort of what we call a one-group pre-test post-design. One group is just the same individuals, and pre-test post-test, because I measure whatever I'm interested in this case, how fit is an individual, before pre-test, time 1, and I measure it after, post-test, time 3. So it's a one-group pre-test, post-test design. Okay, the next design that we can have is actually not about one groups, but about two groups. And that's pretty much now the experiment design that I did with you guys. I had two different groups. But I have a static comparison. So I don't measure something before and after. I only look at something after, but for two different groups, right? So let's say I observe two kinds of individuals. The one set of individuals, they do exercise, and then I look at, after they do exercise, in which shape they are they fit? And I look at another set of individuals who do not exercise, and I look at how fit and in which shape they are at time 3 as well, and then I can compare these two groups with each other. Right. So that's now, if we would put it in another word, or here it's called a static group comparison, but it would be a two-group post-test design. There's no pre-test, but there are two groups. Right? What is the problem with that? Well, it could be that the people who don't do sports to begin with, they are just systematically different. Maybe healthy people tend to do more sports, which makes them even more healthy. You see? That's sort of where now in the ideal world, actually, we would have a two-group pre-test, post-test design. Right? I measure how fit are those individuals before they exercise here, and how fit are those other individuals that don't exercise here at time 1 as well. Then I look at the difference between time 1 and time 3 for this group, and I compare it with the difference from time 1 to time 3 in the second group. That would be a two-group pre-test, post-test design, which is sort of the classical experimental design. It's a two-group pre-test, post-test design. I sort of set up in a slightly different way. I have in the one column the experimental group or the treatment group, and in the other one I have the control group. I compare them on whatever I'm interested in, on the dependent variable, say how fit are individuals. Then to the experimental group, I administer a stimulus or a treatment. Then I remeasure the same dependent variable for both groups. And then I can compare the differences in the differences. So that's much more powerful. So there's much more about these different kind of designs, and there's also, we can complicate things where there's not just one treatment, but there are, let's say, two different treatments at the same time. Then we're talking about factorial designs, and we can talk about full factorial designs, Solomon designs, and all those kind of things. You can read up on these kind of things when you're interested in that. Essentially, these are sort of extensions of these ways of what do I actually compare, right? Where does my treatment come into play, and what am I comparing at the end of the day? Okay, so these are different designs in experiments. Now let's talk a little bit about what I already outlined, what I called Hawthorne effects and solutions to them. Okay, so basically this goes back to our study, which is one of those milestone studies in sociology. It was carried out, it was in the 1920s and 1930s, can't remember. Huge study, Elton Mayo, George Caspar Homans was involved in that. Happened at the Hawthorne Works. Now there was a factory. It's a factory where they actually studied all bunch of things, all kinds of things. Amongst others, they also studied the effect of lighting, I don't know, how well lit is a workplace on the productivity of workers. And what they found was that when there was better lighting, they did an experimental design. Basically, they looked at individuals, they changed the lighting, they gave them better lights, and they looked at how productive are they compared to how productive they were before they gave them a better lit working place. And they found people became more productive. Said, yay, we found something here. Lighting matters. I can make a good argument for that you can work better when you actually see what you're doing. So that's a good argument for that. Said, okay, there we are. But then they went on, and then they did another experiment where they changed the lighting back again. And people became even more productive. I thought, hang on a moment, this can't be true. So what's the point here now? Well, until they realized, and that's what we call nowadays the Hawthorne effect, they realized that the change in individuals' behavior was not driven by the stimulus, by this treatment, by changing the lighting condition, but it was driven by those individuals in this factory feeling important for being part of this study. Suddenly there were researchers running around, looking over their shoulders. They observed them, how they work. Well, if I would stand next to you all the time while you're sitting in the library, you'd better work as well, right? So that's actually what happened here. Yeah? That individuals changed their behavior because of knowing that they were part of an experiment, because they were part of a study. And that explained this fact, that they got even more productive, or they stayed productive when they changed the light back again. Right? Because they were still part of the experiment. But now the experiment was something else. Right? There was still the researcher looking over their shoulders, and that explained this confusing result that they had here. And that's something we really need to think about when we do experiments. We need to think about the fact that by doing the study, by exposing our individuals to, let's say, a laboratory setting where we invite them in, we tell them, now you're participating in an experiment, we might already bias the results. And you see the crux of that, because then at the end of the day, how can you ever find something about the real world, while whenever you study the things that you want to know something about, the very fact that you studied changes the results, right? That's what we call actually low on external validity. That it's difficult to generalize to the population. While internal validity in this context means we are very good with making our causal statement. That this treatment here really affects the outcome, right? Because this is this is our gold standard. This is the only thing that we changed is that. But maybe the real world looks different to that. Right? So that's a problem that we had and that people discovered as they got into experiments more. So this is just a photo from the Hawthorne Works. And this is how you could imagine that they were working. They were doing, I don't know what they were doing there, putting something together. They changed the lighting, switched it on. They looked at how productive individuals are, and they found these differences. In the 1920s, so they found that people work harder and perform better when they were participants in an experiment. How can we deal with that? Well, I already outlined a little bit what we can do about that. We can do single-blind and even better double-blind experiences. Single-blind experience means that I know, as a researcher, who's part of the treatment, who's part of the control group. But the individuals don't know. And to a certain degree, this is sort of, when you think about giving a patient a placebo drug, this is a single blind study. The patient doesn't know. The patient doesn't know if he or she is part of the treatment or the control group. But if the researcher knows, then it's a single blind study. So one side knows about who is in the treatment, who's in the random group. But as I mentioned earlier, this can still introduce a bias from the side of the researcher. Right? Then maybe I'm behaving differently. Maybe I'm giving slightly different instructions to the group that is in the treatment group, because unconsciously I know that this is sort of what I want to find, or maybe this is sort of why I'm behaving differently to a patient, and give other recommendations about doing sports or whatnot, right? So this is where the double-blind experiment comes into play. Right? I think Platoon exemplifies it nicely, where neither the patient nor the researcher knows who is in which group. Right? It's what we call a double-blind experiment, and it eliminates what we call experimental bias. Okay, let me move on. So now after experiments and designs and the Hawthorne effects, now I'm going to talk about, in a little more detail, about 3 different types of experiments. Now, and you already came across 2 of them, well, 1 of them mostly, but the other one, most likely you came across that one as well. And the last one are these quasi-experiments or natural experiments that I'm very fascinated by. Okay, so 3 different types of experiments. We can have laboratory experiments or lab experiments, the field experiments, and what we call the quasi-experiments. Okay, so let's get started with laboratory experiments. All right, so when you, I don't know, maybe before coming to this class, that's what you would have thought about an experiment. There's somebody sitting there in a laboratory doing something, experimenting something, and observing some change, right? While in the social sciences, we can apply that strategy, and a lot of people do. It's actually huge in economics right now, experimental economics, but even in sociology, it's even bigger in psychology and social psychology, right? But then other disciplines pick up. Sociologists do laboratory experiments nowadays as well. Essentially, it's a very controlled experiment in an artificial environment. It's a very artificial setting. And it actually even looks like that. Now, some universities have laboratory settings where they invite people in. They say, I don't know, 50 of you guys can come in to different sessions where the experiment takes place, and then individuals sit in their little cubicles and they are being exposed to whatever the experiment is all about on the computer screen. It's a very controlled setting. It's controlling place, it's controlling time, it's controlling, individuals, who is assigned to which group. It's really controlling all the other things around it, because I know this is what I'm showing the individuals, and all else they see is sort of the world that is a cubicle. So it's a very controlled setting. There's one study, that I found fascinating, because basically some colleagues of mine, not only is it a great paper published in the HAS and the American Journal of Sociology, but also it's a very fun one as well, where they had guys like you test different wines. Often these studies are done with undergraduate populations, right? So it's not a new thing that I'm running an experiment with you guys here. It's sort of what a lot of people do. Well, where else do we have 500 people sitting here, willing to participate, or I don't know. It's also an interesting group to study. So what they did, they had 52 students participating in what they call an test on aesthetic taste in different domains. Right? So they were invited to come in. Actually, they were cheated on. They were told that they kind of, scored particularly well in the exam, but they didn't. So they were just being invited to come in, and they were sitting in their little cubicles. Right? And then they were told that they would interact with 5 other participants in their group. They were always coming in groups of 6s in a way during the experiments. But in fact, they were not interacting with the other 5. They were interacting with some artificial individuals. Right? some fake individuals, some simulated participants. What did they have to do? Well, they're given 3 different wines. Right? So if I get some research money, I'm going to replicate this study. I invite you. Yeah? We'll have some wine. They were given 3 different wines, while in fact all 3 wines were the same. All 3 wines were the same, except that the 3rd one was tainted with vinegar. Okay? So the first and the second one were exactly the same, and the 3rd one was the same wine, but there was vinegar being added. So clearly that was sort of, and they did some other tests before, this was the worst wine. And this is sort of obvious. Okay. So then what happens is that participants, they drink their wine, and then they were given their ratings about the wine. And they were given their ratings after hearing the ratings of these other 4 fake participants before. Maybe you see where this is going, because this is an experiment on conformity. This is an experiment on norm violation and conformity, and at the end about sanctioning of the violation of conformity as well. So these 4 fake ratings all claimed a huge difference between the first and the second wine. Well, in fact, there was no difference. Right? And then the actual individual gave their rating after hearing that 4 other people in this group said there's a big difference between the first and the second one. What do you guess? What happened? Conformity? Well, actually, there was a clear evidence for conformity that 53% of the students then also rated the second wine below the identical first one, although they were exactly the same. Right? So here the treatment was hearing what others say. That's sort of a social experiment. You see how sort of the behaviors of others, now behaviors of others is artificially manipulated through these fake participants. And I tell you that others behave in a certain way. And then I look at what you do. So there was clear evidence for conformity in this study, but on the top of that, there was even more interesting. They also found evidence for sanctioning. So they even started, I'm not going to go into detail about that, but if you want to read up on that, read the article in the AGS. These experiments are very easy to read. There's nothing fancy about it. There's sort of a treatment and a control group here, difference. Sometimes the setting is a little more sophisticated, but still very easy to understand. Very easy to understand, even for I don't know, when you're at the beginning of your studies. So they also found evidence that people started to sanction others who violated the norm. Right? So when somebody actually didn't agree with those 4 fake participants, when somebody said, no, they are exactly the same, and then they had a second round where they actually rated the judgments of the others in the group, people started to sanction those who violated from the conformity. So that's sort of then what we call, the enforcement of false norms or social pressure to conform to a norm can lead those who conform to also sanction others for not conforming. It's a very interesting thing because it can really lead us into a lot of social troubles where we conform to something that actually doesn't happen or is actually not really the true opinion of individuals. On the top of that, although we personally disagreed with a norm or with a certain behavior, we sanction others, we force people to follow that norm. In the literature, it's also known as the emperor's dilemma. There's a story by Hans Christian Andersen about the naked emperor. Everybody says that, beautiful clothes, while in fact the emperor is running around naked, and people start enforcing norms. So anyway, so that's sort of just a little side note here to laboratory experiments, to what I think is a very good and very interesting laboratory studies. The strengths of such laboratory experiments are, while you have a very strong possible evidence for causality, because all you changed was the treatment. Now you sort of manipulated one thing. Laboratory settings allow you to control the situation and take other factors out. And because they are so highly controlled, they are also very easy to replicate. Because there are very clear instructions about how the whole thing looked like. This is sort of how often there are even pictures attached to it, right? About how the situation looks like. Then people can control that. You can replicate results. In this case, we would actually talk about reliability. Yeah? Because if we would do the same thing again, we're very likely to get the same results. What are weaknesses of these laboratory experiments? Well, the setting is often highly artificial. Now think about sitting in this cubicle there, right? And there's somebody, I don't know, there's a screen, and the screen tells you supposedly what the other people in the room say, or what the other people like. This is very artificial. This is not really how the real world goes, right? And that's sort of one of the big criticisms that people have with the laboratory experiments is that, yes, it might tell us about causality, but only in these very specific circumstances of where our experiment took place. That's what we would call, okay, these experiments are high on internal validity. They're really good in getting to the causality, but they're low on being able to generalize to the wider world, because it's so artificial. So that's sort of the big drawback of laboratory experiments. Subjects might be unwilling, can unwillingly bias the results when they know they are part of the experiment. You know, that's sort of the Hawthorne stuff. Or the experimenter can unwillingly introduce a bias with knowing who's exposed to the treatment. So even though the researcher doesn't want to do that, it can still happen. Sample size is often very small. And the last thing that I liked, often these results are done with undergraduate students, and undergraduate students are weird. It's a technical term, you know. It's a technical term, weird. You guys are Western, educated, industrialized, and rich and democratic, right? Compared to the wider population. I don't know, maybe some of you are not rich. But at the very least, if I look at the population here, compared to a random sample of similarly aged people in the world, it's a clear group, right? So actually people started to overcome these shortcomings and go out and, you know, replicate results in, I don't know, somewhere in the Middle East, in India, I don't know. Some colleagues of mine do lab in the field experiments in Uganda, you know, just to see if the group has an effect, yeah. And now we are actually talking about field experiments. And field experiments is really taking the experiments out of the laboratory setting. It's taking it into the field. And remember the kind of stuff that that I talked about, the broken window theories, that was field experiment. These were field experiments, you know, I'm not repeating that. Yeah, it was about these flyers attached to bikes, you know, there was the order condition and the disorder condition. That's not a laboratory setting. That's a real world setting, right? Real people passing by in their real life, and then people study that. What are the strengths of these field experiments? Well, they take place in the real world. You can generalize from these findings because it's not artificial. Weaknesses, well, there could be other things that happen. There could be other things that factor in here. Maybe people who go to their bikes at a certain time when they kind of change the graffiti up there, maybe it's a different kind of people that show up there at different times, right? That's something that now they can't control. They can't control who's showing up at which time. In a laboratory experiment, you can control that. Well, it can be also very time consuming, you know, imagine, you know, they even had to put some graffiti onto the wall, so I imagine that being fun, and being paid for it. It can be subject to researcher bias. There's also the third one called quasi-experiment, but let me talk about that next time. I don't want to rush that, so we'll come back to that next time on Monday. Let me show you the readings. These are the readings for next time. It's mostly about sampling and selection bias. Okay, see you on Monday.